Yes. All
Okay.
good.
So yeah, I'm visiting Michael at the moment in Barcelona, and so I'm downstairs, and I'll give a bit of a presentation and introduction, and then we'll switch over to Michael upstairs in the lab, and they'll do a short lab demo, and we'll see we'll see if things work. Okay, let me get my pointer. Okay, so this talks about live magnetic observation of hyperpolarization dynamics. Um, okay, let's see. So we have here, okay, on this slide, so here's our typical PHIP process. We are incorporating parahydrogen into a molecule, or we have some reversible interaction with a molecule, and we're producing some species, and then we do a polarization transfer. And sometimes that's just done in situ continuously in Saber experiments. And in other cases, this is a separate step. And this polarization transfer can be done at high field or low field, zero field. But this usually involves some sort of sequence of events. So we've got some sort of control Hamiltonian that we're using to transform our parahydrogen derived spin order into some other state. And very often in our applications, that would be polarization on a carbon 13 or maybe a nitrogen 15, something that's a longer lifetime than the hydrogens. And so while this process works reasonably well, and we often get tens of percent polarization in favorable cases, and we have some enormous carbon 13 signal, and we can go off and use that compound for different applications. But we've got this major limitation in NMR in general that we can't observe the spin dynamics during these transfer sequences because while we're pulsing, we're saturating the detector. So you would typically, in an NMR experiment, you're applying a pulse sequence, but then you stop and you observe what's happened. And so here's a nice example of that. Here's a polarization transfer sequence. So we do some PHIP reaction by bubbling in parahydrogen to a sample, and then we drop the field down low and ramp it up. And this is where the polarization transfer happens. And then we shuttle our sample to a high field magnet for detection, in this case, a benchtop magnet. And well, we can do that. And if we want to see how the sequence is performing, we essentially do many experiments. We'll change some parameter and we take many, many data points and we build up a picture. And we often have very good match. Warren mentioned in his talk that the match of spin dynamic simulations to the data is typically very, very good because most of the dynamics are linear. And okay, but this takes an incredibly long time to build up these sort of pictures because you're running the experiment many times over. Um, Okay, so what I'll be talking about today is about doing low field detection because we want to be observing spin systems while we're applying pulse sequences. And if we're doing that at low field, we can't really use inductive detection with a coil because the frequencies are very low. So we need a different type of sensor. And so what's shown here is an alkali metal vapor sensor. This is often called an optically pumped magnetometer. And so this is like a glass cell that contains, in our case, a rubidium vapor, and that's right next to the NMR sample. And here's what the setup might look like. So we've got our sample there with these magnetometers next to it and some coils around it for applying magnetic fields. Okay, and then over here, this is like a little analysis of low field NMR, where we say, well, it should be very simple because the timings are very slow. You don't need to lock the external magnetic field. You don't have impedance effects or susceptibility artifacts, from magnetic inhomogeneity because you're working at very low fields. Um, but on the other hand, yes, it's a relatively low cost type of NMR. But in reality, actually, a lot of these components you end up at a similar cost to a benchtop NMR system. So it's comparable to benchtop NMR, I would say, in the cost and ease of implementation. Okay, so and then well, here's a picture of how a low field NMR experiment might look. So we have a bottle of parahydrogen, and we have our plumbing here. So we are flowing the parahydrogen gas, and that's going to flow into a sample, and our sample's in this 6 millitesla field. Okay, and this is be a classic Saber experiment where we're polarizing protons, in this case in pyridine. So we've got a very typical Saber experiment. And after bubbling for some time in this 6 millitesla field where we're polarizing the pyridine, we bring the sample down into this mu metal shield. And we've got a solenoid here providing some chosen magnetic field. In this case, that would be 10 microtesla. And then we detect. And in the spectrum, which is shown in red here, we measure our pyridine signal, and that's going to be at quite low frequency, 400 or so hertz. Okay, because we're in a very weak B0 field in this case. On the other hand, this is not that far away from, from a high field experiment in terms of what we're observing. This is pulsar quire, and we're detecting nuclear Larmor precession. Okay, so at this point, I'll stop, and if Michael and the others in the lab are ready, they'll give a short demo of this sort of experiment, so you can see this sort of thing in performed in the lab, and you can see the sort of equipment that's involved and how it's done. So I would stop there for a moment and let Michael turn on his camera.
Hello, everyone. Can you give me a quick nod, James, if you hear the audio? This is
You do hear the audio, yeah.
Okay, so welcome to the lab. I echo James in saying that we were unaware that this was a dual presentation. So this is going to be the idea was that we would do a quick demo of the reaction scheme and the magnetometry setup that James just described in his previous slides. And we're going to do this as much as is possible within the constraints of a live presentation. So it might work, it might not work. Let me just share this screen quickly to, can I do that like this? I'll just show you a little bit our type of detection scheme. Do you see that? What do you see on the screen, James?
We don't see the sharing.
Oh, you don't? Okay. Okay, now it needs to be enabled. There we go.
Now it's sharing.
Okay.
This is good.
So this is a window which is showing a live view on our magnetometer, which is sitting in this part of the lab on this table. So there's a magnetic shield, and it's got one of these rubidium vapor sensors inside. And on this side of the lab, we have James's student, Anushka, who's going to do the chemistry part of the experiment. And we'll take the polarized sample from the chemistry experiment and put it in the magnetometer on the other side. So I'll just show you what on the screen right now, what it looks like, what we're going to be doing. So we're going to be applying one of these magnetic field sweeps, and that can be applied on demand. So I can just do it now. And if you look at the top part of the plot, you will see the magnetometer outputs. Okay, here we go. Hopefully that works. Right. So that's moving, and then we see a dip, and then it rises back up again. And that's us doing an on-demand switch from 2 microtesla down to 50 nanotesla, and adiabatically returning to 2 microtesla, which is exactly what should be needed to convert the para spin order of fumarate into magnetization of fumarate. So we're going to try and do that, injecting the sample. Maybe James can explain a bit more about what's going on the chemistry side, and then we'll try and show you what's going on this screen as well. We'll hopefully show you a spectrum afterwards. Okay. So I'll quit the sharing, and then James can tell you what's going on in the chemistry. We'll try and get the camera as close as we can.
Yeah, that would be great. Yeah, if the camera could be a little bit closer, that would be helpful.
Somehow lost the... Here we go. Yeah.
Okay. Temporarily turn off my video. Okay, so over there they've got the sort of a typical PHIP simple experiment with a parahydrogen bottle on top of the table and some hand-operated valves. There we go. Okay, so there's some hand-operated valves there. And what Anusha's doing right now is she's just got the sample, which is our precursor material, in an NMR tube, and she's bubbling parahydrogen through it. And that's all just being done in an oil bath. And the oil bath's at about 120 degrees, so we get this nice and warm. And, well, that speeds up the reaction. So she's just been warming up the sample there, and now she will start bubbling. And she'll do this for around 60 seconds or so. So something that we measured the other day actually is we were measuring the temperature inside these samples during this bubbling, wondering whether the parahydrogen bubbling itself is cooling down the sample. I mean, the oil bath's at 120 degrees, And we want our sample to be at a similar temperature. So the solvent is water, but under pressure, it can heat up higher than 100 degrees. Um, and well, we essentially measured that actually bubbling parahydrogen through the sample, the cooling is by less than 1 degree. And presumably that's just due to the very, very low specific heat capacity of hydrogen gas. So that's actually not really something we need to worry about in these experiments. Okay, and so right now she's bubbling parahydrogen into that sample, and we have this reaction occurring. So essentially all those fumarate molecules are being formed with the protons in the singlet state, and that's close to an eigenstate in Earth's magnetic field. And so, well, essentially the molecules are relatively happy in that state. The lifetime is around 60 seconds. So it's a reasonably long singlet lifetime for those protons. And now she will quickly open up that tube, put a syringe in there, and extract the sample into a syringe. And now what she has is this singlet polarized fumarate in the syringe. And she's gonna go over to the zero field chamber, where there's a line there where she can connect up the sample and inject it into the spectrometer. And then Michael on the computer will press now apply the pulse sequence, and that will run. And then inside that magnetic shield is the magnetometer that's detecting the signal. Okay.
So we were hoping for some success here, and we were rehearsing this throughout the afternoon and basically getting some bad results. But we'd be pleased to know that we have got some signal and we can show you. Let me try and share the screen again. I just have to click that. Okay. Here we go. So this is a Fourier transform signal here, and it's absolutely huge. It's a little bit broadened actually. So if we wanted to see this nice and clean, we would see it. Yeah, you see it dies away kind of quickly. Actually, this is filtered for the wrong range. Excuse me. Yeah. Let's try and see the time domain signal here. Okay, so it dies away pretty quickly. Which is perhaps because of the inhomogeneity of the magnetic field that the sample is present in. But we've also seen effects where the magnetic field of the sample itself causes broadening of the resonance. This is simple dipole field effects. I don't know if James is going to talk about those in the rest of the presentation, but this is basically the end of the lab demo. So I hand back to him anyway. And
okay.
Yeah, well, maybe we return here for the questions. See you later.
All right, thanks very much.
Right. Let's.
Well, I didn't ask. Are you guys still showing something, or that's all?
So I will just continue with the presentation now, if that's all right.
Please go ahead.
Okay. And you can see my screen again?
Yes.
Perfect. Okay, so I'll carry on then. So, all right. So now back to slides. So this is a picture of what was really going on in there in that experiment. So this is a magnetic shield here, and there's a solenoid that passes through the shield, and there's a sample inside. Okay, so what they did is they injected the hyperpolarized sample into that sample chamber, and there's the magnetometer here next to it, and that's what you're using to detect the sample. And so in that sort of experiment, the reason we have this geometry, and here's a nice rendering of that, if this solenoid passes through the magnetic shield, then when you turn on a current in that solenoid, you're producing a field inside at the sample, but the field outside the solenoid is almost zero. And a few minutes ago, I ran a quick console simulation of that just to illustrate this quite nicely. So here, this is our mu metal shielding, and there's a solenoid passing through. And inside this solenoid, I've just set the current to provide 100 microtesla field. And you can actually see in the norm of the magnetic field that the field lines will pass through the magnetic shielding and come back around. And here, where the magnetometer would be next to the sample, there's almost no magnetic field. And in reality, the factor of difference we typically get in a solenoid like this is about a factor of 1,000. And what that means is you can apply microtesla fields to the sample, but your magnetometer is only seeing nanotesla fields. And that means that your magnetometer can remain sensitive while you're applying pulses to the sample. Okay, so in this sort of experiment, then we can try some different things. This is the one we were hoping to show today, but we just had some problems throughout the morning, and so we didn't quite get to this point. This is the experiment we were hoping to show. So in this one, well, we start out with our fumarate in this proton singlet state, and that's a non-magnetic form of the molecule. And well, it's in a high magnetic field, it's in Earth's field, and we inject it into this low field system. And what we're going to do is we just drop down the solenoid field to 400 nanotesla. And this 400 nanotesla corresponds to an avoided crossing between two states. So if we drop the field to this matching condition, we get coherent oscillations between these two states. And it turns out, well, if so we're essentially the magnetization will, or the population will oscillate into a state that is magnetic. And so if we just detect with a magnetometer, we start with zero magnetization, but then it builds up and then decays again, and we get these coherent oscillations. And we see that for fumarate. We see this nice signal that oscillates up and down, and it has a decay time constant of about 22 seconds. And we can then Fourier transform this signal. This is like a zero quantum coherence. And what we see is we see a peak at about 2 hertz, and it's a relatively narrow signal. Okay, if we then do the same experiment, but we fully label the sample with carbon-13, so now, you know, we're essentially, we fully label with carbon-13, we're getting about 50 times higher magnetization from that sample, because instead of just 2% of the molecules participating, because it's symmetric, we now have 100% of the molecules participating in the spin dynamics. The magnetization is significantly larger, so we can scale it down a little bit in this red plot, so they're sort of comparable. But what we see is actually that the coherence decays significantly faster, and then we have this this zero frequency component that also decays, and we Fourier transform that, and we see what we have instead of this nice sharp peak is a broad lump with some interesting shape. And so we think there's something different going on here, and that's that the large magnetization of this sample is slightly disrupting the spin dynamics. If this sample is very highly magnetic, it means that we are no longer sitting at this avoided crossing, we're shifted away somewhere. And that depends on sample shape and polarization of the spins. Okay, well, all right, that's good to know. So we'll come back to that thought in a moment. So here then is our here's the depiction of the experiment again, and we can do different types of experiments. We can do another type where we drop the field down and then ramp it up, and that's what Michael showed in that demonstration. We drop the field down, ramp it up to 2 microtesla, and by doing this, we should also create polarization of the spins. And if we track that live, so we can see this in real time, we see the magnetization starts close to 0, and then it jumps up as we pass through this 400 nanotesla region. Okay, so we see the magnetization jump up, and this is very well behaved for this non-labeled sample. And we can do it with a different molecule that has different J couplings. So it's an isotopolog of this, it's an isotope of this fumaric acid. Sorry, isotopomer. So the J couplings are different, and that means that the spin dynamics are going to be very different. And we see that clearly. The magnetization buildup profile looks totally different to that from fumarate. But it's still very well behaved, and it still matches simulations, the simulation shown in gray. So again, if we haven't isotopically enriched the samples, we can very well mimic what's going on with our simulations. Okay, but over here, this is the experiment that I showed before. Over here, then, let's look at another one where we're also ramping up the magnetic field to get polarization transfer. In this case, if we do it with 99% carbon labeling again, now again we run into the same problem where the experiment does not match simulation. So in this experiment, we're inverting the magnetic field, And we think our magnetization should increase it's a sort of a complicated profile, but it's shown in gray what we expect to happen. And that's clearly not what's going on. So we've got this, we basically see that at these high spin densities and high polarization values, we have these nonlinear spin dynamics that don't match up with simulations. And we probably need to modify our simulations to account for the sample's magnetic field. And so that's shown here then. So we can run these sort of experiments. So if we want to know, well, what's the magnetic field inside the sample? I've put some numbers down here so we can just see some clear numbers. So if we ignore geometric demagnetization effects, then we can calculate the magnetic field from some different samples. So if we have 100 millimolar of protons at 50% polarization, they should produce a 530 nanotesla field. This is clearly large. This is very, very large. This is larger than our level anti-crossing field. And even water, these internal magnetization effects have been shown extensively, and it's a problem with water as well. You get shifts on the order of 1 hertz or so just from the magnetization of the water sample at high field. And so we see these effects in our experiments. If we're looking, so over here we see a sample that is not, okay, let's look at this one here. So in gray, we see a sample that's at 2% carbon-13 enrichment. And essentially our signals are at these 2 frequencies and they don't change over time. So this is the first scan. The signal decays as we scan over longer time periods. The signal decays, but there's no real shift. But then if we fully label the sample, we do see a shift. And that shift, well, we measure it, it's about 3 hertz. And it means our sample has a 70 nanotesla internal field that's causing a resonance frequency shift. Okay, well, 70 nanotesla is not devastating, but it's still of the same order of magnitude as our level anti-crossing field. And so we think clearly this will have an effect on our polarization transfer. And that was shown very nicely in a paper from last year by Larry Nasdagis. He showed in that work that you can do these polarization transfer experiments, but as you increase the concentration of your sample, at low concentrations here, the polarization that you generate is sort of flat, is as expected, and you can get up close to 50% proton polarization. But as you get to higher and higher concentrations, meaning that the total magnetization in the sample gets larger, then the polarization that you're creating decreases. And over here, where you're at 1 molar concentration of the material you're trying to polarize, you only have 3% proton polarization. And that's probably due to this effect where the internal magnetic field from the sample is disrupting the transfer sequence. Okay, so and if you're interested in this, there's a sort of a solution to this problem in the paper using dipolar decoupling sequences. So you can read more about that here. And so in summary, then we've got a nice way now to probe this interesting regime where the high magnetization of samples causes nonlinear spin dynamics, and we're able to see those things live during the transfer sequences. So we're hoping that there's sort of an interesting area to do research in. Okay, and with that, I'd like to conclude and say thank you to the members of my group at Jülich and to these external collaborators who contributed to this work, and especially Michael, who's hosting me and Anushka for the week at ICFO. Okay, thanks very much for your attention.
Okay. It was great. We have time for one short question, and there's a question in Q&A from Malcolm Levitt. Do you see a prospect of real-time adjustment of pulse sequence in response to the real-time detected magnetization?
Yeah, okay. So that's a question about control, I guess. That would be like a real-time feedback control where you're feeding back the signal from the detector to the pulse sequence. It seems like that sort of thing should be possible in these simple experiments where we just have the magnetic field in one axis. We just have like this one axis magnetic field, and if the spin dynamics that we're seeing they're occurring on a time scale of tens of hertz, And that's slow enough that, yeah, I imagine that you could essentially have some processing of the signal and iteration of the transfer sequence and feed that back. And Michael's put up his finger, so he wants to add something.
My short answer to that question, I think it was from Malcolm, but I didn't hear the audio very well, is yes, why not? And it's something that should be easy to implement.
Yes, for sure. I mean, I would also use this opportunity to say that recently we implemented the same geometry of the experiment, right? Where you have a piercing solenoid, and that allows you to really decouple these two things, measure the signal from the sample without affecting the sensor. And as long as delay is permissible, or some small delay, I think it should be possible.
So well, it doesn't necessarily have to be a feedback at the frequency of the spin transition. Okay, it could be that the frequency of the spin transition depends on field, and then all you have to do is slowly change the field in order to keep the spin transition at the right frequency, for example.
All right, let's thank the speakers again for the wonderful.
Thanks, everyone.